It should allow, maybe if it's an amoeba cell membrane, it should allow food to come in. It should allow water to come in. It should allow ammonia to escape, CO2 to escape, etc. So that means that boundary which is present is not merely a physical structure, but it also has to serve certain functional roles. So those functional roles become very important. So when people started seeing, can we view the cell membrane under an electron microscope? They were actually left dissatisfied, because even under an electron microscope, you find that we are unable to make out what exactly are going to be the components present in a cell membrane. Is it like a plastic sheet that is present as one unit? Is it going to be more of a boundary, like how you have a brick wall where you actually take individual bricks, but you are able to create a unit? How is the cell membrane going to be? So there was so much of curiosity, and we had a number of scientists who gave their own theories as to what might be the cell membrane structure. For example, we had the scientist Robertson, we had scientist called as scientist group called as Danieli and Davson. Each one of them, they proposed their own idea of what the cell membrane is going to be. But there were a lot of demerits or drawbacks to the model that they proposed. And for the first time, we had two scientists, that is Jonathan Singer and Garth Nicholson, in 1972. They conducted a lot of cell membrane analysis on the RBCs of human beings. Why did they take the RBC of human beings in order to understand the cell structure, and not why not take simply cheek cells, or why not take bacterial cells? I'll just come to that a little bit later. So they worked on the RBCs of human RBCs, and using that they were able to have a lot of chemical analysis, that is qualitative analysis, quantitative analysis, and using that information that they got from qualitative and quantitative analysis, and looking at the input that came from the electron microscope studies of cell membrane, they were able to propose a model which is presently, even after more than 4 decades, it is presently the most accepted model to explain the structure for cell membrane, and that model is called as the fluid mosaic model. I will go ahead and I will explain what exactly is this word fluid. What is the word mosaic? Why are these words used by Singer and Nicholson in order to explain the cell membrane structure? So I will come to all these points which I have promised you. Why take RBCs? Why use the words fluid mosaic model to explain the structure? We will be looking at all those components one by one. So let us go ahead and see what were scientists able to understand when they analyzed cell membranes. So when Singer and Nicholson analyzed the cell membranes of RBC, let us try and understand what they were able to get. So firstly, when they did a qualitative analysis, they found out these are going to be some of the components of the cell membrane. And what components were present? So before we go to the assembly of the components, let's look at the components of the cell membrane. So the components which were present in a large amount, okay? So a large amount of the cell membrane is going to be made up of lipids. Lipids, as you are aware, they are non-polymer biomolecules. Okay, they are not polymers, they are non-polymer biomolecules. So one of the biggest components present in cell membranes was lipids. Finger and Nicholson had analyzed other cells also, like cheek cells and bacterial cells, certain plant cell tissues, etc. And they found out that the percentage of this lipids is going to vary not just from the cells of one species to another species, but also between cells of the same organism. Between one cell of one kind and another cell of another kind, there is going to be a lot of difference in the percentage of lipids that are present. So let us not get too worried about certain facts, what is the percentage, etc. That of course, students can learn up on their own. So let's not worry about that. So lipids definitely are going to be one of the components. What are the types of lipids that are present inside the cell membrane? So the lipids are going to be of two types. The first and the most predominant type, or the majority type lipids that are present, are called as phospholipids. So one of the predominant type of lipids that are present in cell membranes are called as phospholipids. Okay, now phospholipids, I am sure students have, you all have learned considerable amount of biochemistry under the chemistry subject. You all have also learned about biomolecules under biology, and you are aware about what phospholipids are, right? Now, phospholipids are what? They are special type of glycerides. Phospholipids are going to be special type of glycerides. Now, what do I need to make a glyceride? To make a glyceride, I basically require a molecule of glycerol. Okay, so that's the first thing that I need in order to make a glyceride. So I will require a molecule of glycerol. Okay, that's going to be a molecule of glycerol that I have. Okay, along with a molecule of glycerol, what is it that I require? I will require, in order to create a phospholipid, two fatty acid molecules. So I will need two molecules of fatty acid. Then by using a dehydration reaction, or you can call it as a condensation reaction, that is by the removal of a molecule of water, I am going to attach the fatty acid to one of the carbons here, to this OH group. Fatty acid has got a COOH group. And glycerol here, you can see it has got three OH groups present here. So I can utilize this OH group and use the COOH group of the fatty acid, and I can join it to one part of the glycerol. Another fatty acid molecule is going to be joined to the next OH group here by a condensation reaction. Once I do that, the last OH group which is present is not going to react with a fatty acid molecule to create a phospholipid. Instead, it will join phosphoric acid. That is a phosphate group. Okay, so I'll repeat again, right? This is basic knowledge that you all are supposed to have after learning biomolecules. So I have a glycerol molecule, and two of the OH groups will react with fatty acids here, and then one of the OH group will react with phosphoric acid. So what happens when you have that situation? Let us say I'm going to join this. I'll just show it in a diagrammatic way. Okay. Now I will not show it in as a structural formula here. For example, let us say if I have, this is going to be my glycerol molecule which is present. What am I going to have? I'm going to have this kind of a situation. So I'm going to have, this is going to be one fatty acid molecule that is present. This is going to be another fatty acid molecule that is going to be present. And then at the end, I'm going to have a phosphate group which is going to be seen. These are two fatty acid molecules, glycerol molecule and the phosphate group. So what happens as a result? This whole structure which is present, this entire structure which is going to be seen, is going to be a polar part of this molecule. So for this phospholipid, this area which is seen is going to be polar in nature. Or we can say that it likes to be with water. It doesn't get repelled by water. So we call this part as the hydrophilic part of the phospholipid molecule. These are going to be fatty acid chains, and you have the hydrocarbon part which is going to extend because the COOH group has got attached to the glycerol. These two parts of the fatty acids, chains which are present, that is part of the phospholipid, that does not like water. Our part, and we call it as the hydrophobic part. As a result, the entire phospholipid molecule becomes amphipathic in nature. The whole phospholipid molecule becomes amphipathic in nature. The whole thing will be amphipathic in nature. One polar and one non-polar part is present. Now, obviously, we cannot draw a phospholipid molecule in this way when we have to represent a cell membrane. From now onwards, this phospholipid molecule which is present, I am going to be representing it in this particular class simply as this. That is, I'm going to be showing this as the hydrophilic head, which has that glycerol and the phosphate part present. This is the hydrophilic head. And I'm going to be calling this as the hydrophobic tail. The hydrophilic part is polar. The hydrophobic tail is going to be non-polar in nature. That is first component that is present, and it is the predominant component that is going to be seen as part of lipids. Okay, what is the next part that is going to be seen? The next type of lipid that is seen may not be seen in all cell membranes or across all the living organisms. Some living organisms may show absence of this lipid. Some of them may show the presence of this lipid, etc. And that lipid is going to be cholesterol. This is good cholesterol, okay? This is not bad cholesterol. This is good cholesterol, which gives certain very specific properties to the cell membrane. So cholesterol is also one of the lipids that is present in cell membrane. So the first component, I hope, is clear. Let's go to the next component. So the next component that is present in the cell membrane after qualitative analysis was found to be proteins. So the next component that was present was found to be proteins. What are the various types of proteins that are going to be seen? You have predominantly the proteins that are present in the cell membrane of organisms are going to be globular proteins. They are globular proteins. Okay, so they are going to exist in the tertiary structure. Some of them can exist in the quaternary structure also. So they are going to be globular proteins that are seen. Is it possible that some fibrous proteins are also seen in the cell membrane? Definitely, it's going to be possible. So you will find small amounts of fibrous proteins that can be seen inside the cell membrane. So proteins that are present in cell membrane can be globular proteins and fibrous proteins. We are going to be learning how to classify these proteins on the basis of their location. So I will be coming to the location. Here I'm only describing the ingredients. ಈಗ ಒಂದು ಮಸಾಲೆ ದೋಸೆ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಏನೇನ್ ಸಾಮಾನ್ ಬೇಕು. I'm only listing the ingredients. I have not told you how to put them together. We'll come to that. So based on the location, the protein types are going to be of different ways or of different types they are going to be seen. We'll come to that a little bit later, right? So proteins can be seen. What else can be seen in the cell membrane apart from this? So the cell membrane will also show the presence of small amounts of carbohydrates. So small amounts of carbohydrates are also seen. What type of carbohydrates? Will there be glucose? Will there be sucrose? Will there be starch, etc.? No. The types of carbohydrates that are present in cell membranes belong to a very specific type of polysaccharides that are called as oligosaccharides. They are called as oligosaccharides. Okay, oligosaccharides, oligo, the word oligo itself means very small. Oligo andre minute or small it means. So oligosaccharides are going to have limited number of monomers present. So whatever is the monomer, there will be limited number of monomers, usually maybe between 3 to 7, sometimes maybe between 3 to 10 monomers can be seen, right? Those are going to be called as oligosaccharides. Like disaccharides are going to have two monomers, no? For example, maltose is a disaccharide. It's got two molecules of glucose. Sucrose is a disaccharide. It's got a molecule of glucose and a molecule of fructose. Like that, oligosaccharides will have limited number, not hundreds of molecules like starch or glycogen or cellulose. Limited number of monosaccharide molecules will be present between 3 to 7 and sometimes maybe up to 10. That is what we'll find. So these are all the components that are going to be seen. Now let us see how to assemble them together. Now going ahead, as I said, qualitative analysis gave Singer and Nicholson an idea as to what is present. Right? For example, I know that if I have to make dosa batter, I know I have to have rice. I know I have to have some dal, some methi seeds, etc. I must also know what is the proportion to have, right? So immediately when Singer and Nicholson and the team conducted quantitative analysis, how much of lipids are present, how much of proteins are present, what they realized is now already your simple microscope has already given us an idea what is going to be the size of a human RBC. So a human RBC, we know its size, we know its width. When you look at a human RBC, okay, this is how it is going to be. It's a biconcave disc-like structure. When you see it from the top view, it is going to be circular in nature. And when you see it in a cut view, this is how it will appear. That's going to be a human RBC. Human RBCs are going to be exactly 7 microns in diameter. And we also know what is the value for the narrow part, what is going to be the value for the broad part. So we know exactly its circumference. We know the surface area size. Okay, I'm sure in geometry you all know that a three-dimensional object, if we tell you what are the dimensions, you will be able to calculate the surface area. So when you look at the surface area of the human RBC, the lipids appear to be 2 times. The lipids appear to be 2 times than the surface area of the RBC. Immediately that gave an idea that lipids have to be present as a bilayer. Okay, so lipids have to be present in 2 layers. That immediately the scientists were able to decipher. If they had taken any other cell like cheek cells or WBC or parenchyma cells, there is no standard size for those cells. Therefore, it is difficult to find out what the surface area is. Whereas for human RBCs, because of that specific size, we are very clear that we were supposed to get this amount of lipids. Now there is double amount of lipids, which gives you an idea that there is a bilipid layer that is going to be seen, right? So that's why they took human RBCs. Let's go ahead and try and find out how exactly ಈಗ ನಾನು describe ಮಾಡಿರೋ components ಇದೆಯಲ್ಲ, how are you going to put them together? Okay, let us try and describe this. When Singer and Nicholson were asked by a group of scientists and press people who were asking them, how do you describe your fluid mosaic model? Then very clearly Singer and Nicholson had a wonderful way of describing. They said cell membrane, imagine cell membrane has got lipids and then has proteins and small amounts of your carbohydrates. So what they described it as, they said, imagine that the lipids are present as a sea or an ocean. The proteins are present as icebergs in that ocean. That is the way in which, or the analogy that Singer and Nicholson gave. They said that if the lipids are present as a sea, the proteins are present in an embedded manner within it, right? And I think, you know, it is wonderful as students of science for us to realize that every cell of every organism is covered by a layer which actually is made up of individual molecules, but the layer is formed only because they are sitting together. Okay, it is like I want to create a compound wall stand next to one another. That itself is giving me the compound wall. It's not one unit. There are individual components that are present, but the way they are assembled, the way they are put together makes it a membrane, right? So let us try and understand about the cell membrane structure. I will be giving you now a two-dimensional diagram as to how the cell membrane structure is going to be. Let us try and understand how it is going to be. So I'll draw it and I'll also keep explaining. So firstly, what we have is we are going to have the lipids are going to be arranged in a bilayer. What do I mean by bilayer? Please understand, even terrestrial organisms are going to have every cell having the environment as aqueous and the cytoplasm is aqueous. So though we have become successfully terrestrial creatures, our cells love to be in contact with some amount of moisture around them, some amount of water environment is needed for the proper structure and working of every cell. So we are not able to become, a cell is not able to become completely terrestrial that way, or when you look at in an evolutionary context. So every cell is, its home is water based and its environment is also going to be water based. So what happens as a result, you will have the lipid molecules are going to be arranged in this way. Okay, so the lipid molecules are arranged in this way. This is one layer of lipids that I have. This is going to be the next layer of lipids that are present. Why am I drawing it this way? As I had spoken to you earlier, I'm showing each phospholipid molecule as a head with the two fatty acid tails that are present. So the tails are two fatty acid chains, and then this is the head which has got the glycerol with the phosphate part. Now, this, let me imagine this to be the environmental side. So I'll consider this part to be the environment side. Environment side may be actually the abiotic environment. For example, imagine there is a euglena present in a pond or an amoeba present in a pond, then the environment becomes the pond, or it might be the tissue fluid for an organism. Okay, some fluid that is present outside. Imagine maybe one of your smooth muscle cells. So the tissue fluid becomes the environment. So environment side, this is going to be the environment side. What I'm having this side is going to be the cytosol side. That is the cytoplasmic side. Okay, this is what I'm going to consider. This is environmental side and cytosol side. So what I'm going to have, I'm going to have these lipid molecules which are going to be arranged in a bilayer. That is, the heads face the aqueous environment side, the heads face the aqueous cytosol side, and the tails, which are hydrophobic, will face one another because they are okay. This is not lipid based, the water based, this is not water based. They are okay to be facing one another. That is how I'm going to have these arranged here, right? So I'm going to have these structures present in this manner, and a bilipid layer is going to be created here in this way. Okay, right? So I'm going to draw some of these layers present in this manner. This is going to be the lipids that are present. You can imagine it as this way. Okay. For example, imagine I have taken hundreds of table tennis balls, and to those balls I have put two straws, and I have arranged a lot of these table tennis balls with all the straws facing the room. And then what I've done, I've taken another set of table tennis balls with straws and I have placed it over them. That's how the structure is going to be. So this is how they are going to be arranged, right? These are all the various lipids that are going to be arranged in this manner as a bilipid layer. This is the bilipid layer that I have present. Okay, so I have drawn it in a 2D manner. Please understand that, right? I will be showing you a PowerPoint at the end of this presentation to try and understand how it is right? Okay, so this is how the lipids are present. So what is the percentage of lipids that can be seen? Of course, it is going to vary. You look at the cell membranes of animals that are present in Arctic oceans, very cold regions. They will have, the percentage of lipids to proteins will definitely vary considerably. You see a tropical animal, it will vary. So depending on the environment, the percentages, so there's no fixed number that this many only lipids or proteins should be. It can be varying. Okay, that is one thing. Let's now come to the proteins. As I told you last time, there are two types of proteins, globular proteins and fibrous proteins. Now, first, let us come to the globular proteins. How are the globular proteins arranged? So the globular proteins are of two types. I'll repeat again, the globular proteins are of two types. Some of them are embedded within the bilipid layer. Such proteins which are embedded in the bilipid layer are going to be called as integral protein or intrinsic proteins. So those which are embedded in the bilipid layer are called as integral proteins or intrinsic proteins. Of what kinds are they going to be? For example, let me draw some of the integral proteins. You can see I have drawn this integral protein here. This is one integral protein that is present here. Okay, it is going to be embedded within the bilipid layer. So I told you about the table tennis balls that I have arranged. No, balls and straws upside, and then the straws this side and all the balls that are present. So between them, I make a gap and I put a protein. That is one of the integral proteins that I have shown here. I will show another integral protein here. This is another integral protein that I have drawn here. Why have I drawn it that way? I'll come to that. Okay, this is another integral protein. I can show one more integral protein in this manner. Okay, that is another integral protein. I can show one more integral protein this way. That is one more integral protein. So in different ways, I can draw these proteins. I can show another one here. Okay, these are all different types of integral proteins that are going to be seen. As you can see, they are all going to be embedded in the membrane. Some are going to be embedded in a bitypical manner. That is, they are embedded throughout the bilipid layer. See this one, it is embedded throughout the bilipid layer. This is embedded throughout the bilipid layer. This is not only embedded throughout the bilipid layer, it is going to be also extending or projecting outside on both sides. This is also present across both the lipid layers. Whereas this one is a monotypic protein. It is present only across a single lipid layer. But whatever said and done, whatever I have drawn for you now, they are all going to be integral proteins, which are either partially embedded or completely embedded and emerging from the bilipid layer. They are all integral proteins. If you have to remove them from the membrane, it is very difficult. You need to have high speed centrifugation technique done in order to remove them from the lipid layer. That is what you're going to find here. Okay, next let us come to peripheral proteins. So what are peripheral proteins? Peripheral proteins are not embedded within the lipid layer, but they are present outside the lipid layer. For example, I can draw this as a peripheral protein. This is one peripheral protein. This is another peripheral protein. So I can have peripheral proteins which are going to be present outside the bilipid layer. Of course, they are very few, but still they are going to be seen. Peripheral proteins are also called as surface proteins because they are present on the surface of the bilipid layer. Next, what am I going to have? These are all the various types of integral proteins that can be seen. Next, what are we having? Next, we'll come to the fibrous proteins. Which is the most common fibrous protein seen in cell membrane? The most common one happens to be actin. Actin fibers. I'm sure you have heard about actin and myosin in the context of skeletal muscles or muscles as in general. So muscles in general show contractile proteins called as actin and myosin. Muscle cells are not the only places where actin can be located. Actin also acts as cytoskeleton for the cell and is also present very close to the cell membrane. So sometimes you can have actin filaments or called as microfilaments which can be present in order to attach one component of the protein to the other component of the protein in this manner. So you can have one component and another component attached to one another by means of actin filaments. One point that all of you should note is actin filaments are always present towards the cytosol side. They are not seen towards the environment side unless the cell membrane is covered by a cell wall. So if it is not covered by a cell wall, they are always existing towards the cytosol side and not towards the environment side. We'll come to the functions of the actin proteins also in a little bit while. Okay, let's come to the next component. So I'll come to the next component. Next component is you can find the presence of some cholesterol also. I'm showing it as a as a ring here. So that is just, I've just used this. Please remember, this is just diagrammatic. It is not it is not the exact representation. So cholesterol molecules also can be present here and there within the cell membrane. That is what you're going to have. Right. Let's come now. So what is the last component that is going to be seen? The last component that is present will be the carbohydrates. Where are carbohydrates seen? Never seen on the cytosol side. Carbohydrates are always present on the environment side. And what type of carbohydrates? They are going to be oligosaccharides. Where will they be attached? Where are they present here? Oligosaccharides can be either attached to the head of a phospholipid molecule. It can be either attached to the head of a phospholipid molecule or it can be attached to a protein. It can be either attached to the head of a phospholipid molecule or can be attached to a protein, right? That is going to be the oligosaccharide that is present always towards the environment side. If a protein is attached to a carbohydrate, that is an oligosaccharide, this entire unit which is present is called as a glycoprotein. The word GLYC refers to a carbohydrate. So if you have one of these oligosaccharides attached to a protein, then you call that entire protein as a glycoprotein. In the same way, if a carbohydrate is attached to the phospholipid molecule, you will call that entire structure as a glycolipid. Right? Now let me do a quick recap of the structure. Then I'll come to why it's called as fluid mosaic model. And I'll also come to what are the functions of what we are able to see here. Though the topic today is just cell membrane structure, but I'll just take maybe a few minutes to explain to you the functions also what we are looking at. Because please remember, you know, for example, now I'm sure you all have heard that Watson and Crick, James Watson and Francis Crick, they were able to give us the structure, of double-stranded DNA. They called it as the double helix. Now, when somebody proposes a structure, it's not just that I can give any structure and forget about it. No, when I when I propose a structure, the structure has to suit the functions that molecule or that part has to perform. You know, it's for example, if I have to propose a structure for how a car is, it's not just anything that I can go and sit in. It should also serve the purpose of how it's going to move, right? How it's going to go uphill, how is it going to park itself, how is it going to reverse, right? In the same way, when Singer and Nicholson proposed the structure of the cell membrane, it also has to satisfy the functional components. You know, it has to say that yes, the functions are also going to be explained by this model. So we'll come to fluid mosaic model. First, we'll do a recap quickly. So what I have is I'm going to have a bi-lipid layer that is present. These are the lipids that are going to be seen. As I said, it's going to be a 2D structure here. In a 3D structure, what are you going to have? For example, let us say these are the lipid molecules that are present in this manner. Okay, let's say those are the lipid molecules present in this manner. That is how the structure is going to be. That is how the structure is going to be. And then these are present here. These are going to be located here. Correct? That's how the structure is going to be. So it is present, you know, in a 3D manner. That's going to be the bilipid layer that is going to be seen. And embedded within the bilipid layer, you have the proteins. So the proteins are going to be integral or intrinsic proteins. If they are partially or completely embedded in the membrane, they can even project out of the membrane. The proteins which are present on the surface and not embedded, we call them as surface proteins or peripheral proteins. These surface proteins and integral proteins are all globular proteins, whereas you also have small amounts of fibrous proteins associated with the cell membrane, and that's going to be represented by microfilaments, that is actin filaments, which are present always towards the cytosol side. That is what you're going to find. Okay, next we also spoke about how oligosaccharide chains, that is carbohydrates, can be attached to some of the parts of the cell membrane, and they are always present on the environment side. If they can be attached either to a protein or to a phospholipid molecule, in which case this protein will be described as a glycoprotein and the lipid molecule will be described as a glycolipid molecule. And we also said that depending on the organism, etc., you can have another lipid that is going to be seen within the cell membrane, which may be cholesterol, good cholesterol, which may be present as part of the cell membrane. That's going to be the structure. Now let us try and understand about what all these components are going to be. I've already explained to you about why the bilipid layer is present this side. This is water side, this is water side. So the heads are hydrophilic, they like water. So they are polar parts, so they are present towards the water side. The tails are hydrophobic parts, and that is they are non-polar parts which face one another. Okay, this is what you have. Now what are you going to have? Let's come to some of the functions that you have seen here. Right now, for example, firstly, uh, let us look at this protein. Okay, now what you find in this protein is, suppose from this side, I want a gas to pass from this area to this area. Maybe it is going to be oxygen, it may be carbon dioxide. If it is a plant cell, it would love carbon dioxide to come in so that it can reach the chloroplast, correct? So I'm going to have some gas passing. Now what happens is a gas can simply diffuse through this membrane without taking any help from any of these parts. It can simply diffuse through, right? So that is going to be a gas which can move through. But all the components which come from one area to another area are not going to be only gases. Okay, for example, water probably has to enter a cell, glucose has to enter a cell, right? So these are some of the components that have to come in. Some components have to leave, etc. Insulin, for example, has to come out from the beta cell of pancreas and has to go out, etc. So you have so many things that we have moved, or there is movement that is of a unique nature. So what has to happen? Sometimes, suppose there is, let us say, a lipid based substance. For example, let us say estrogen, which is a steroidal lipid soluble hormone. So estrogen or progesterone can easily get dissolved through the lipid layer and come into the cytoplasm. not an issue at all because it is lipid soluble. You know, even if there were no proteins and let's say there was a membrane made up of pure lipids, it is still able to move through. That's not a problem at all. But let us say some other substances which are not lipid soluble. Okay, or which may be charged particles like sodium ions, for example. Now, those substances which are not lipid soluble, those substances which are very large molecules, or substances which are charged particles, they can't simply go through this. They will need a proper door or a tunnel to pass through. And that is offered by this kind of a protein, which is going to be called as a channel protein. I have drawn a channel which is open. You have different types of channels. I'm sure you have heard in the nervous system about voltage gated channels and normal channels that are present, or leak channels, etc. There are different types of channels seen. So this is a channel protein. I have drawn it in this manner. Please remember, what I've drawn it in this manner is basically going to be present as you have these long beads, no? Through which you can pass a wire, like that it is present. So a lipid, let us say you are offering an area for a substance to pass through without having to meet the lipid hydrophobic area. For example, water has to pass through. So you have water channels present because water can't pass through like that. So water has to pass through a channel so that it doesn't get affected by the hydrophobic tail area of the lipid bilayer. So this is a channel protein which I've shown as a cut section. So that is also seen. Right? So some of these can be channel proteins. Some of them are carrier proteins. What does it mean by a carrier protein? For example, some of these proteins are present in such a manner that let us say some substance comes and gets attached here. This protein is able to turn itself and deliver the substance on the opposite side. Okay, for example, this way. Let us say I'll draw another protein to help you understand what is going to be a carrier protein. So a carrier protein may be present like this. For example, this way, you may have a protein present like this. Let us say. So what this protein is able to do here, this protein can actually meet a substance. Suppose, I want to deliver this particular molecule. It can get attached to this protein. This protein can turn and deliver this on the opposite side. So such type of carrier proteins are also seen, right? This is how all the various proteins are present. So remember, the proteins are present in a manner that they serve for the transport of certain substances. It may be as a channel, it may be as a carrier, etc. Apart from that, do the proteins have any other roles? Some of these proteins can also be enzymes. They need not be only physically present there. Some of them are also enzymes. You have a number of membrane-based enzyme proteins that are present. So some of them may have such enzyme role also. In addition to that, glycoproteins particularly act as antigens. That is, suppose your WBC is roaming around in the body and it meets a liver cell, it meets a kidney cell, it meets a stomach cell, etc. It should be able to say, this cell belongs to me, I will not attack it. Correct? How does it identify this cell belongs to its own body? It is able to identify by badges, like how you wear a badge when you go to college. In the same way, cells also have badges, identification badges are present. Which is the identification badge there? That's going to be this glycoprotein. Okay, other parts also can be identified, but glycoproteins particularly act as antigens. They are going to be the badges that a cell has to help in cell to cell recognition. Okay, so those are going to be glycoproteins that are seen. Okay, now next I'll come to, why do I require these actin filaments? In order to help you understand why actin filaments are needed, let us come to the understanding of this word, of this term that we use for the cell membrane called as fluid mosaic nature. What does that mean? Let me first explain the mosaic part. All of you have seen mosaic floorings. The tile floors sometimes are going to be mosaic floors. One tile does not resemble the next tile, and the arrangement of all those colored stones is in a random manner. There is no fixed manner in which they can be arranged. In the same way, the arrangement of lipids and the proteins is very random, right? The arrangement of this is going to be very random. Therefore, you call this model as the mosaic model because of the random arrangement of the components. It's not going to be the same way all the time, you know, so it is going to be random arrangement. So your liver cell membrane is going to be very different from your own body's muscle cell or from your own body's neutrophil, right? So that is random arrangement. That's why it's called as a mosaic. Why is it called as fluid? Here, the word fluid does not mean liquid. The word fluid here means flexible. It means it has got the ability to change itself. For example, I have these components present, no? This component which is present here, or let's say this component which is present here, can move itself around the lipid layer. For example, this component can shift here and these lipids can occupy its place. If I want, I can destroy this component and replace it with lipids too. If I want, I can add one more oligosaccharide chain somewhere. So what we mean by fluid here is that it's not liquid. It means it's quasi-fluid. They say quasi-fluid means like fluid, right? It means that it has the ability to modify itself. That ability or that flexibility allows it to undergo phagocytosis, to undergo microvilli formation, to allow for secretion of substances, to allow for pseudopodia formation, etc. Otherwise, or to produce endomembrane system, etc. So the fluid nature allows for modification, for physical modification, for chemical modification, and so on. That's what the name fluid mosaic nature means. Now, apart from the components, as I said, this component can move around. It can come from here to here, etc. You imagine it is like you have a tub of water and there are small little icebergs that are floating. So you can make those icebergs move from one area to another, correct? In the same way, they can all interchange positions. But apart from that, there may be certain positions that probably can be moved, but they have to be kept together as a unit. For example, let us say, ನಿಮ್ಮ ಮನೆಯಲ್ಲಿ ಇವಾಗ ಒಂದು ಬಾಗ್ಲಿದೆ. Okay, ಈಗ summer ಅಲ್ಲಿ you will get wind, you will get a lot of breeze from the west side. So you want more windows to go towards the west side. Now, obviously, when you build your house, you can't make that arrangement. ಎಲ್ಲಿ window ಇರುತ್ತೋ ಅಲ್ಲೇ ಇರುತ್ತೆ, correct? But let us say you had a home where you can actually move the windows around. ಇವಾಗ ಇಲ್ಲಿ ಬೇಕು ವಿಂಡೋ ಇಲ್ಲಿ ಹಾಕೋಣ, ಅಲ್ಲಿದ್ದ ಗಾಳಿ ಬರ್ತಾ ಇದೆ ಆ ಕಡೆ ವಿಂಡೋ ಹಾಕೋಣ ಅನ್ಕೊಳ್ಳಿ. ಇವಾಗ ನೀವು ವಿಂಡೋ ಮೂವ್ ಮಾಡಿದಾಗ you have to move the door, the rods of the window, the frame of the window, the glass of the window. Everything should be moved as a unit, right? You can't move them separately. In the same way, even though flexibility is needed, sometimes some of the proteins may have you may have cooperative function. For example, this protein and this protein may have some kind of functions which are going to be codependent or which are interrelated. So though I can move this, when I move this, I want this also to move in the same manner. For that purpose, I can keep both of them attached by actin filament. So if this moves, this will also move with it. That's why I need these actin filaments to be present, right? So that's going to be your fluid mosaic model. As I said, the idea behind today's lecture was not to go into the functions of the membrane into detail. We are not speaking about facilitated diffusion or active transport or what is simple diffusion, etc. So that was not the purpose. It was mainly about cell membrane structure. Okay, I will just share a presentation for all of you, and with just, I don't have more slides, just I think 2 or 3 slides are present, and we will be able to see the cell membrane structure here. So just a few slides as a quick recap for you. Okay, that is Jonathan Singer and Garth Nicholson, who in 1972 gave us the fluid mosaic model. This year will be the golden jubilee of the fluid mosaic model, right? And fluid mosaic model, you can see this is just one of the images that I just downloaded. So you can see a number of various types. So all the reds are the phospholipid heads that are present. All the yellow tails you can see. You will also be able to see all the blue color structures that are present, which are present in the form of a number of various shapes they can be seen in, sizes they are seen. Some are channel proteins, some are going to be, non-channel proteins are present, etc. Some of them will show the presence of oligosaccharide chains on the outside. And internally, you can also have the cytoskeletal elements, that is actin filaments can also be seen inside. So this is just the 3D version of how the cell membrane looks like. It is very, very amazing. I think over the past maybe 4 decades or so, almost all research that happens in the field of medicine, in the field of just general biology, molecular biology, it has to do so much with cell membrane, because that's what gives us so much of identity. As I explained earlier itself, you just need two things to have metabolism. That is, you just need cytoplasm and you just need cell membrane. And that itself, just individual components which are behaving as such a wonderful unit together, and they behave in one integrated manner, right? You can see here, just another GIF image, which is able to show how the water molecules can actually pass through the water channel proteins. You can also see how the artist, the animator has shown how the Bi-Lipid layer is going to be present, etc. Right? So with that, I come to the end of this particular lecture. It has been a pleasure giving this lecture to you. Thank you so much. Over to Umesh sir. Sir, you are mute. You are mute. Ah, yes. Yes, sir.
Thank you very much, ma'am. In
Thank you.
fact, it's indeed a great presentation. With in the end, we can see the visuals also.
Yes,
With these
thank you.
visuals, it will be very easy to understand the students also.
Yes,
Now
sir.
I
Thank you.
request,
Thank you, sir.
thank you very much, ma'am. Now I request students and teachers, if any questions, they can raise their virtual hands so that we can unmute them. They can speak to directly to the madam, or they can mention in the chat box. Any questions, please do mention. I think there are no questions as of now, ma'am. In
No
future, if,
problem, sir.
yeah,
Yeah.
the teachers and students can approach us anytime, and they can mention their questions in the feedback link as well as in our website, and they can follow our all website and all social media channels. And for upcoming program details also, we will be updating daily. I request all the students to subscribe and give information to other students, classmates, or any science aspirant students or teachers, whoever it may be. It will really be helpful for you in the last CET exams or anyway. Thank you very much, ma'am.
Thank you. Thank you
Uh,
so much, sir. Thank you. Thank you so much. Thank you.
Before winding up, I would like to give vote of thanks on behalf of KSTA and on behalf of all the students and teachers First and foremost, I would like to thank Dr. Lakshmi Murthy ma'am for today's lecture on cell membrane. ಇವತ್ತಿನ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಿ ಎಲ್ಲರ ಜ್ಞಾನ ವೃದ್ಧಿಗೆ ಕಾರಣರಾಗಿದ್ದಕ್ಕೆ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ಮತ್ತೊಂದು ಸಾರಿ ತಮಗೆ ವಂದನೆಗಳನ್ನು ಅರ್ಪಿಸ್ತೀನಿ.
ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್.
ಅಲ್ಲದೆ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದ ಪ್ರೊಫೆಸರ್ ಎಸ್ ಅಯ್ಯಪ್ಪನ್ ಮತ್ತು ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದ ಡಾಕ್ಟರ್ ಎಂ ರಮೇಶ್ ಸರ್ ಅವರ ಅವಿರತ ಮಾರ್ಗದರ್ಶನ ಮತ್ತು ಬೆಂಬಲದಿಂದ ಎಲ್ಲ ಕಾರ್ಯಕ್ರಮಗಳು, ಈ ರೀತಿಯ ವಿಜ್ಞಾನದ ಉಪನ್ಯಾಸ ಸರಣಿಗಳು ತುಂಬಾ ಸುಲಭವಾಗಿ ಹಾಗೂ ಸುಲಲಿತವಾಗಿ ನಡೀತಾ ಇದ್ದಾವೆ. ಅವರೆಲ್ಲರಿಗೂ ತಮ್ಮ ಪರವಾಗಿ ಹಾಗೂ ನನ್ನ ವೈಯಕ್ತಿಕ ಪರವಾಗಿ ತುಂಬ ಹೃದಯದ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸ್ತೀನಿ. ಅದೇ ರೀತಿ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಮತ್ತು ಶಿಕ್ಷಕವೃಂದಕ್ಕೆ ಈ ವೆಬ್ಎಕ್ಸ್ ಪ್ಲಾಟ್ಫಾರ್ಮ್ ನಲ್ಲಿ ಮತ್ತು ಯೂಟ್ಯೂಬ್ ನಲ್ಲಿ ನಮ್ಮೊಂದಿಗೆ ಉಪನ್ಯಾಸ ಆಲಿಸಿದ ಎಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸ್ತೀನಿ. ಹಾಗೆ ನನ್ನ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರಾದ ಡಾಕ್ಟರ್ ಶ್ರೀನಿವಾಸ ವಿ.ಕೆ. ಸಿದ್ದೇಗೌಡ, ಆಮೇಲೆ ಶ್ರೀ ಸಿದ್ದೇಗೌಡ ಅವರು, ಆಮೇಲೆ ಆಂಟನಿ ಲೋಬೋ ಅವರು ತಾಂತ್ರಿಕ ಸಹಾಯ ನೀಡಿರೋದಕ್ಕೆ ಅವ್ರಿಗೂ ನನ್ನ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತಾ ಇವತ್ತಿನ ಉಪನ್ಯಾಸವನ್ನ ಇಲ್ಲಿಗೆ ಮುಗಿಸ್ತಾ ಇದ್ವಿ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
Thank you so much. Thank you so much.